Good morning to everybody and welcome to our new episode of Itanong Mo kay Sir Pat.
Like a Hellcat.
Thank you to the school's division of Tarlac Province for making this crash course possible. Special thanks to Dr. Pozon, Dr. Linsao, and Madam Palafox. Throughout the discussion proper, the participants are expected to first, please turn your screens into video modes so that I could clearly see you. Prima facie. Thank you so much. Second, give me the floor throughout the discussion proper. Questions may be asked after the presentation. Third, please take down notes. Fourth, in this webinar, each participant is expected to submit their research proposals. Please refer to the issued Division Memorandum number 124 Series 2020 for the deadlines to beat. Lastly, this is not an advanced course. Only basic concepts will be covered in our discussion. The main objective of this webinar is to at least help the beginning researchers to prepare their action research proposals. Our foci are the beginning researchers who are classroom teachers, regardless of their ranks and their action researches. In this episode, we are going to talk about the basic statistical methods of analyzing data. Particularly, we will focus on the basic intervals for part 1, the basic sampling techniques for part 2, and the basic statistical procedures for part 3. Without much ado, let's get started. Intervals are used in interpreting the results in a rating scale instrument. The 3-point, 4-point, and 5-point scale intervals were commonly used. The intervals are presented as follows. In as much as possible, try to minimize the use of even point scales like the 4-point scale because of some statistical concerns attached to it, especially to survey type and perception-based studies. Tend to use odd point scales like the 3-point and the 5-point scales. If adjectival options are few, use the 3-point scale. If there are more adjectival options, use the 5-point scale. When results are intended to be used later on for correlational or comparative purposes, use the 5-point scale. Moving forward, let us discuss the part 2, basic sampling techniques. In a nutshell, statistics is this branch of mathematics that deals with the collection, presentation, interpretation, and analysis of data. Statistical process always involves 4 stages. Stage 1, this is the collection of data. Stage 2 is the presentation of data. Stage 3 is the interpretation of data. And stage 4 is the analysis of data. This is the gist of the entire statistical story. For the part 1 of our discussion, we will deal on the basic sampling techniques. We will peel off the skin of the stage 1 of the statistical process, which is the collection of data. This is the preparatory procedures leading to the actual collection of data. Specifically, let us dig into the population types, the eligibility of criteria, the sampling theory, the sampling criteria, the sample size, and the sampling techniques. In doing an action research, the population is usually our source of data. When we talk about population, we refer to number 1, people. Number 2, they should be present in one place or institution. Number 3, they should share common characteristics. Having that said, let us have a quick analysis. Example number 1. Assuming that you are the master teacher in a large school. You like that? You surely like that. You are the master teacher in TLE. Your school is truly large. that your school managed to have goats in your school farm. Yes, your school has a goat farm. But one day, your goats got ill. As the master teacher in TLE, you were held responsible in solving the causes of the illness of the goats. You will examine what they eat, what their poops look like, etc. Assuming that you have 25 goats, what is your population size? How many are there in your population? The answer is 0. Why? Because you do not have a population. Remember that whenever we are talking about population, we are referring to people. Goats are not people, so there is no population.
Yay!
Okay, example number 2. This time, you are the ICT coordinator in your school. Your computer units in the computer lab have been hit by a certain virus. As the ICT coordinator, you were held responsible in solving this problem. Assuming that you have 28 units in the lab, what is your population size? How many are there in your population? The answer again is 0. Why? Because computers are not people. Remember that whenever we are talking about population, we are talking about people. If they are not people, there is no population. Okay? So this time, let us have an example that has a population. Let us talk about the people in an elementary school. When we talk about the people in an elementary school, we are actually talking about two populations. The pupils are considered one population and the teachers are considered as a separate population. They are both people, they are located in one place or institution, but they do not share common characteristics. The characteristics of the pupils are not the same as the characteristics of the teachers. So the pupils compose an entire population and the teachers compose a separate population. However, population cannot be as easy as that. It could be possible that there is a population within a population. Going back to the population of pupils, all the pupils in that school is considered as one entire population. But within the population of pupils, we have grade 1 pupils, grade 2 pupils, grade 3 pupils, and so on up to the grade 6 pupils. Obviously, the characteristics of grade 1 pupils are not the same as those in grade 6. Therefore, the group of grade 1 pupils is considered their distinct population, and the group of grade 6 pupils as another population. We call that case as stratified population. As we are talking about a population within a population, a strata or stratum is a segment of a population. In this case, grade 1 pupils compose of a segment of a population, grade 2 pupils as another segment, and so on. When we talk about the target population, we are pertaining to a group of individuals who meet the criteria that we need in our study.
Thank you. Thank you.
Once the population is clear to us, sampling refers to the process of finding the samples. And what are the samples? These are the individuals who are chosen to be part of the study. They are the subsets of the population. The samples compose the fewer version of the entire population. Let's have this example. There are 30 employees in a department. only 5 were chosen for a random drug testing. What's the population size? It's 30. And what is the sample size? It's 5. Very good. The eligibility criteria are the characteristics determined by the researcher, to define who should be included or excluded from the population. This is simply the characteristics of the population that we need in our study. Examples are gender, age, marital status, education, and so on. The sampling theory refers to the mathematical way of effectively acquiring a sample who would accurately reflect the population under study. To make things simpler, sampling theory tells us that the more, the better. Even the sample size is fewer than the population size. Increasing the sample size closer to the population size makes everything better. Remember that talking about sampling theory, the more, the better. This implies that in your action research, assuming that there are only 8 students who are qualified to be your respondents, sampling is not applicable because 8 is only a small number in the first place. So get them all. Include all those 8 students in your study. Also, sampling theory assumes that the characteristics of the samples must be reflective of the characteristics of the entire population. Whatever the results are using the samples must be assumed to be the truth in the entire population. A sampling unit is the specific location which can be used during sampling process, while a sampling frame is the complete list of sampling units from which the sample is drawn. For the steps in sampling, first, identify the target population. However, you cannot do this unless you specify the criteria for selection. How do we specify the criteria for selection? Return to your statement of the problem. Third, identify the sample size and specify the sampling design. Lastly, recruit the samples. Let me give you a few moments to copy the steps. Here's the catch. Assuming that we have a roasted pig. As a researcher, you have a question that you want to answer. Let's say that your question is this. Is this roasted pig delicious? To answer this, you obviously need to taste it. But the issue is this. Are you going to eat the entire roasted pig just for you to find out whether it is delicious or not? Of course not. Instead, you are only going to cut a small portion of the meat and then taste it. Remember, titikim ka lang. If that small cut is delicious for you, you will assume that the entire roasted pig is delicious. In the course of statistics, the entire roasted pig is your population. The small cut that you tasted is your sample. So let's have everything clear when identifying your needed number of respondents. Gaano ka ba kalakas tumikim? In statistics, that is not easy to answer. But let's see how far we could simplify things as we get further. Sure enough, you have an idea how to compute the sample size. We usually compute the Slovan's formula. Yes, it's pronounced A. Slovan. We have Borg and Gall, the Cochran's formula, the concept of 30%, and many more. Sampling is a complex thing. In fact, If you would examine the prospectus of the MS Statistics program of UP in Diliman, you will notice that sampling is a 6-unit endeavor. But we will not dig into the details. Despite the many ways of computing the sample size, here in the Schools Division of Tarlac Province, let us limit the use of Cochran's formula whenever possible and applicable. The statistical discussion is the statistical discussion on its justification will not be tackled through this platform. When talking about sampling, we will use the Cochran's formula. And here is the Cochran's formula. Our sample is denoted by the lowercase n. It is the adjusted sample size that we are going to use, regardless how large or small the population size is. But we could not compute for the lowercase n without computing first the n sub o. Z is the abscissa of the normal curve. You will use 1.96 for the value of z. But if your study is experimental, z will not anymore be 1.96. P is the proportion of an attribute. Q is 1 less P. E is the desired level of precision. You will use 0.05 for the value of E. But if your study is experimental, E will not anymore be 0.05. The uppercase N is your population size. So let us study how to use the Cochran's formula. Let us have ICT integration to simplify the process. Let us use Excel. Follow this pattern. Type your data on exact cells. Otherwise, the trick will not work. On cell B1, type the population size. On cell B2, type the proportion. On cell B3, type the value for 1 minus P. On cell B1, type that exact formula as it is. Don't miss anything, not even the number of parentheses. So be careful. After typing the formula, hit the enter key to see for yourself the sample size. To tell you honestly, the most difficult part in the use of the Cochran's formula is the value of P. It is the proportion. To be sure, find a statistician and be guided. But if you cannot find a statistician to guide you, use P equals 0.5. That's your last resort. Now that you have already computed using the Cochran's formula the needed number of respondents in your study, let us now talk about the actual ways of selecting them. In a 100-sized population, assuming that you only need 15, 1-5, how do you pick those 15? Here's how it goes. Sampling techniques. Sampling techniques are the ways of choosing the samples. There are two major categories of sampling techniques. One is the probability sampling, where every person has the equal chance of being selected. Two is the non-probability sampling, where, unlike probability sampling, not everyone has the equal chance of being selected. One probability sampling is called simple random sampling. It is also called as lottery technique, fishbowl technique, and snowflakes technique. Simply put, simple random sampling means draw lots, bunutan. We also have stratified random sampling. This is used when dealing with stratified populations. We have talked about stratified populations already. Every sample is selected from each subpopulation or stratum. For the non-probability sampling, we have what we call as the convenient sampling, wherein we choose our respondents because of their proximity. Since they are already near us, they will be chosen conveniently to participate in the study. Another non-probability sampling is the purposive sampling. It is also called as judgmental sampling, selective sampling, and subjective sampling. In this kind of sampling technique, respondents are chosen by purpose. They must possess all the meticulously crafted characteristics that are needed in the study. Usually, purposive sampling is used when the required characteristics are only manifested by very few people. Let's try this. Let us test ourselves a bit. A survey will be given to 100 students, randomly selected from the freshman classes at XYZ High School. What is the population? The answer is all the freshman classes. They are people. They are located in one place or institution. They share common characteristics. Another. Mrs. X samples her class by draw lots. Which type of sampling method is this? The keyword is draw lots. So it should be simple random sampling. And so we're done talking about the part 1 of our discussion, the basic sampling techniques. Let's proceed to the next.